Okay, welcome to Biology 515. This is Human Physiology 1. And we're looking at nerve impulse or action potentials. So, how a nerve works. Basically, pretty important. We send nerve impulses every day, every second. Thousands of things need to be controlled. Okay, and all your nerves work pretty much the same way. Okay, whether we're talking inside the brain or inside the peripheral nervous system. Okay. Today we'll just look at the nerve itself. We'll look at the differences between them tomorrow. In terms to know, hopefully you got some of this prior to, okay? But the main one we're going to focus on is the axon terminal. Okay? The other terms are dendrite, a glial cell. Okay, we'll look at one specific glial cell today, but understand glial cells are support cells. Okay? Otherwise, the actual function of the action potential, we're going to need to know basically the top two. We're going to need to understand sodium-potassium pump and membrane channels and gates. So these are all parts of a nerve membrane. And basically to send a nerve signal, what we're going to do is we're just going to control the passage of ions. And by controlling the passage of ions, we're going to create a concentration gradient, an electrical gradient, or electrical chemical gradient is specifically what it's called. Okay, we're going to get a high to low concentration. And this is going to give us a negative charge on our membrane. Okay, and basically the reversal of that charge of depolarization is what's going to be what we call an action potential. Okay, if you refer back to diffusion across the membrane, okay, that would be the main process we're going to look at here. Okay, all right, here we go. Here are our terms. Axon, like I said, is our main focus. That's how we send signals in a nerve. Okay, so every nerve is going to have two main parts, a dendrite and an axon. Dendrite is how it receives signals. So if it's a sensory nerve, okay, meaning that it brings senses in from the body, okay, that nerve is going to, like I said, touch receptor in your hand. Okay, there's going to be a bunch of dendrites there that feel touch. Okay, they're going to stimulate a reaction, okay, which sends a message down the axon. That's basically what we're going to look at in the action potential. Okay, so axon is big, long cables. Okay, so you have nerve signals that run for, or nerve cells that run for feet long, thin cables. Okay, so almost microscopically thin. But they run all the way from like your finger to your spinal cord, or from your toes to your spinal cord. Okay, so they have to be able, or the main part of the body is the axon, or the nerve cell body is the axon. Okay. Dendrite receives signals, axon sends, support cells are glial cells. Okay, so nerves only work, including your brain, have to work in specific chemistry. So they have to be able to maintain this concentration of electrochemical gradient, specifically potassium and sodium. There's some other ones, there's some chlorine, there's some other things that go in there. And these support cells are constantly making sure we have that specific chemical gradient. They're actually running all the things that should be done with a cell, because we know nerve cells don't divide after they reach maturity. So we have these support cells that basically keep them functioning. Then we're going to look at one specific glial cell, that's a Schwann cell. Okay. And then the parts that we're actually going to look at, sodium potassium pump. So the main thing about sodium potassium pump is it is active transport. Okay, so if you recall active transport, across the membrane means one, we're using energy, and two, it can go against that electrical chemical gradient. So it can go from low position to high position by using energy. Okay, that's going to be really important. In order to establish a gradient, okay, we're going to have to use energy to pump against the gradient. Okay, the second one are these protein channels. If you recall the membranes, okay, they're mostly made up of phospholipids, and phospholipids are impervious to charged or ions. Okay, so we have these protein channels, we call them gates. And we're going to look at two of them, a sodium and a potassium gate, okay, which are going to work by basic diffusion, movement from particles from high to low. Okay, so these proteins allow for those charged particles to pass. Okay, and lastly, depolarization. Depolarization just means a changing from positive to, or excuse me, from negative to positive in this case. Okay, it would work the other way as well. But basically, we're going to set a negative charge on our membrane, and we're going to depolarize that to positive, and that's going to be our action potential. Okay, so we look at a nerve cell body real quick. Dendrites are how we receive information. You can see they're spiraled out there to the top left. Okay, axons are how we send that information. And the axons are going to be this big, long, tail-like looking structure of the nerve. And they'll finish at the axon terminal, which can connect to multiple different sites. Okay? So whether we're talking peripheral or nervous, okay, we want to be able to activate multiple pathways. Okay? You can see that the axon is covered in this blue material. Okay? Typically in your body, it's going to be white. Okay? And this is a protein called myelin. Myelin is a fatty protein. Okay? Why do we cover our nerves with fat? Well, fat's an insulator. Okay? So it prevents that electrical charge from escaping. Why do you cover rubber, or why do you cover an electrical cable with rubber? Pretty simple. Okay? So that one, people don't get electrocuted. So why do we cover it with fat? We don't get electrocuted. But the main reason is that it locks in that impulse, so we don't lose that charge to anything else. So in an electrical cable, that rubber keeps the electricity going to where you want it to go rather than losing some into space or into air, okay, which does conduct some electricity, hence lightning. Okay, same thing in your body. We want to really focus that charge so that it's going to where we want it to go. Okay, that's the main purpose. It also does speed up the transmission. Okay, so the actual nerve impulse or action impulse can jump through these little gaps called the node of Ranvier. Okay, so if we look at it here, okay, nodes of Ranvier, okay, are these little gaps in that myelin sheath, and the nerve impulse can actually jump on those, but that's something for another day. Okay, all right, action potential. How long does it take? About a millisecond. So if you look at when we actually start that action potential, okay, from 1 millisecond to about 2 milliseconds here, okay, we have our action potential or our depolarization. Okay, so it takes about a millisecond. Okay, it takes a little bit of time to actually stimulate the nerve, so we'll call it 2 milliseconds. Okay, but once we actually decide we're going to go, about a millisecond to actually send the information. So pretty darn fast, 1/100th of a second. Okay, now it takes about 3 more milliseconds if we look at it here, from 2 to get it back to our point. It's about 3 milliseconds. Okay, where we get able to send another signal. That's really important because we don't want our brain to be confused or our muscles to be confused, okay, where that signal gets compounded upon each other. Two big things here. Number one, it's an all or nothing. Okay, we send the same electrical signal every time. It goes from negative 70 millivolts at rest to positive 40 when we're actually doing our action potential. That does not change. So whether you poke your finger with a little pinprick, okay, or you cut it off, we're still sending the same signal. Okay, so how your brain interprets that is the number of signals, not the power of each signal. Okay, so you can see here we have some failed initiations. That's just simply not enough of a stimulus to actually send that signal. So your nerve does require a certain level before it actually sends that signal. Okay? All right, here we go. Parts of the membrane, we're looking at the sodium-potassium pump and the sodium and potassium gates. Okay, so the main thing here is that we're looking at active transports. Okay, so this sodium-potassium pump here in the middle, okay, is what's going to create our chemical gradient. Okay, so remember again, ions cannot pass through the membrane except for at these membrane proteins. And these membrane proteins are specific. So they're only going to work for one specific ion. Okay? So sodium can only go through the sodium gate or get pumped out through this sodium-potassium pump, and potassium can only leave through the potassium gate and enter through that sodium-potassium pump. Okay? So essentially what we're going to do here is we're going to use active transport to grab all of these sodium molecules and move them outside the cell. Okay, so we're going to get all these sodium ions out here. At rest, okay, we'll look at the actual terminology. Okay, but we're going to get all these sodium ions at exterior, okay, and we're in the process, we're going to pump all the potassiums inside, okay. We're going to let potassium kind of leak out, okay, through basic diffusion. Remember, diffusion does not require any energy, okay, to create our concentration gradient. So let's look at the actual process. Here's the sodium-potassium pump. So you see it's a 3:2 ratio. So we're going to take 3 sodium ions out and 2 potassium ions in. Okay, you can see that only works when they all combine. Okay, and again, this is using energy. All right, so establishing a potential. Okay, so here's the basic 3 things we're looking at: sodium-potassium pump, potassium gate, sodium gate. Okay, so at rest, you're using energy. That's the big one. Rest, you're using energy. Okay, so we're going to use our sodium-potassium pump, actively pushing sodium out and potassium in. Okay, sodium gates are going to be closed. So all that sodium that's getting piled up outside, all these purple ions here, okay, are going to get trapped into this extracellular matrix. Okay, and meanwhile, potassium gets pumped inside, and we're going to open up that potassium gate to allow these potassium ions to naturally flow out. Okay, you can see them color-coded here. Okay, so due to the excess positive charge outside, that makes our internal structure negatively charged. Right? So to send our action potential, all we got to do is flip that charge. Well, flipping that charge is pretty easy. What we're going to do is we're going to shut off that pump, so no more energy being used. Okay, and we're going to allow those sodium gates to open. Okay, and meanwhile we'll close the potassium gates as well. So it's going to keep that potassium level constant. But because we created basically a 0 concentration of sodium inside the cell, all that sodium is going to come flooding in instantly. That's why we used all that energy at rest, was so that we would trigger this instantaneously. Like we looked before, it's about a millisecond to go. Okay, that's because all that sodium wants to go inside that cell through basic diffusion. So it doesn't actually require energy to send your action impulse. Okay, and as that sodium crosses through that cell membrane, okay, it's going to change that negative charge into positive, because all of our sodiums are positively charged, okay, into a positive 40 millivolts. Okay, and this is going to happen in sequence down the axon. So if we segment the axon here, okay, so it's going to happen here. We're going to trigger the first sodium gate, okay, then it will go 2, okay, and then so on and so forth down that axon terminal or axon until you reach the axon terminal. Okay, the axon terminal is then going to release neurochemicals to stimulate the next nerve. Okay, so you can see it here on the little chart. We're going to depolarize section by section here in green, and as one section is depolarizing, the previous section is repolarizing. Again, the repolarization step takes about 2 to 3 milliseconds. Okay. Last but not least, we got that little bit of a gap there, that hyperpolarization. Okay, so we got to reset that nerve. That means turning that sodium-potassium pump back on, okay, which is pumping that sodium actively out of the cell. We're going to have a temporary step where our nerve is extra negative because all that sodium is being pumped out, okay, faster than the potassium is being pumped back in. So we're going to get that little hyperpolarization state where the nerve cannot fire. Okay, That's big, okay, so that your muscles or your brain or whatever the next neuron is can interpret signal after signal. Okay, simple idea is a cramp. We've all had cramps in our muscles. Okay, a cramp is simply a problem with that hyperpolarization state where you're not getting that potassium back into the nerve. That's why you eat bananas to eliminate cramps and drink water. Is so that we can get that potassium back in to reset that nerve. Otherwise, it's like over tapping that button on your gaming console where you're just hitting it so fast that you're essentially spamming that button. That's what's happening in a cramp. Okay. All right. Hope you enjoyed Nerve Impulse. Questions, please ask. Okay. Thank you for your time.